Good morning. Today, our group will be discussing on microarray, but more specifically on protein microarray. In this video, we will be discussing on the mechanism, application, as well as classification of protein microarray. In protein microarray, it consists of a support system such as a glass slide, bead, nitrocellulose membrane to which an array of capture proteins is bound. Prop molecules, usually labeled with fluorescent dye, are added to the array. Hence, any reaction between the prop and the immobilized protein will emit a fluorescent signal that is read by a laser scanner. This technology is also considered as a multiplex device, as it can be miniaturized to contain tens of thousands of capture features arranged in a grid, each specific to a protein. This gives an advantage to isolate as well as study many potential biomarker proteins simultaneously. On the basis of its application, protein microarrays can fall into three major classes, which is first analytical, second functional, and lastly reverse phase protein microarrays, which will be discussed furthermore in this video.
Analytical protein microarray involve the use of fractions, tissues, or cell lysats in analysis. In general, this array is developed through the immobilizations of purified protein on glass slides. It can be further divided into analyte labeled assay format and sandwich assay format. The first format requires only one antibody that acts as protein capture agent and is typically being referred as antibody capture. Meanwhile, sandwich assay format involves two antibodies, which one of it functions to immobilize targeted proteins on a solid path. The second antibody is a reporter whereby they send signal for protein detections. Although both of these two types shows different sensitivity and specificity, it plays important role in measuring the protein expression.
Functional protein microarray. Functional protein microarray is also known as target protein microarray, which is an important tool for high-throughput analysis of proteomes and other large collections of proteins developed by immobilizing large number of purified proteins. This enables study of various biochemical properties of protein, such as binding activities. The principle behind this array is that the array protein must maintain the original conformation so that meaningful interaction can occur on the array surface. The advantages of controlling the precise mode of surface attachment through the use of appropriate affinity tag are that the immobilized protein will have a homogeneous orientation, resulting in higher specific activity and higher signal-to-noise ratio in assays with less interference from non-specific interactions. One of the examples of the application is to array proteins and use them to probe for antibody present, such as would be found in serum, or for cross-activity of the antibodies.
The term reverse phase refers to the fact that the analyte antigen is immobilized as a capture molecule rather than immobilizing an antibody as the capture molecule. The RPMA has been successfully applied in the cancer studies, especially in the analysis of the state of pro-survival, apoptosis, and mitogenesis pathways within microdissected human malignant lesions, including carcinoma or host trauma. The RPMA also applied in the investigation of serial studies to predict your therapeutic response with imaging and molecular analysis, which is known as I-SPY1 and I-SPY2 for patients with stage 2 and 3 of breast cancer.
The application of this technology is basically revolves on the microarray's key features miniaturization and parallelization. The first one is substrate fingerprinting. One of the main applications of microarray in proteomics is to map ligand binding specificities of a protein. A comprehensive knowledge of enzyme substrate specificity can help the successful design of highly potent and selective inhibitors, facilitating the drug recovery process. Other than that is ligand or inhibitor binding. It allows protein to be labeled with a fluorescent dye and then incubated with an array of small molecules. In conclusion, protein microarray have evolved from being used primarily as a basic analytical research tools to now viable options for more complex application in proteomics.